Okay. Hello, everyone. It's a pleasure to be here at the African Society of Human Genetics, uh, even if it is virtual. And it's my pleasure to introduce Professor Anangu Chaudhry, who obtained his PhD in bioinformatics from the University of Calcutta, India, and had his training in Sydney Brenner Institute for Molecular Bioscience. And then he's currently employed as a senior scientist and co-leading several components for the NIH-funded the Africa WITS in-depth partnership for genomic studies that is also co-chairing the H3Africa genome analysis working group role in several major genomic studies conducted in Africa, including South African Human Genome Program and the H3Africa Genotyping Array Design and the H3Africa Whole Genome Study. His research interests as population genomics and genetics and genomics of complex traits, and he's going to give us a very interesting, I'm sure, presentation about genetic variation, migration, admixture, and selection, a topic that's of interest to all of us. Please go ahead, Professor Chaudhry. Professor Chaudhry.
Hello, everyone. Good morning, good afternoon, good evening, depending on wherever you are. So, I would like to begin by thanking the organizers for this wonderful opportunity of sharing some snapshots of the exciting population genetics work that I have been associated to by virtue of being a part of the H3Africa Consortium. So, let us start with a simple question. How different is my or say your genome from the 3 gigabase reference human genome that was published in 2001? So although the exact number would probably vary, but on an average a genome differs at 4 to 5 million sites. That's a A will be replaced by a G or a T or an insertion or a deletion from the reference genome. So therefore, in terms of percentage, less than 0.02% of the genome that we would normally sequence will be different, while the rest of the 99.8% plus would be exactly the same as the reference genome. However, as you know that it is this small percentage that contains all the critical information, such as what will be the color of our skin, eye, whether we will have predisposition to certain diseases, whether we will be able to digest milk when adults, or we will be able to be benefited by particular medicine, and so on. But what is more interesting to us as population geneticists, that in addition to all this information, a genome is also kind of a time capsule. So it condenses the evolutionary history of all the ancestors and can provide answers to questions about them, such as whether they migrated or not, whether they met and exchanged genes with other populations, what was their population size through time, and also gives us some idea on the diseases and pathogens they might have encountered. However, a single genome can only provide limited information. As you can see in this plot, each of us harbor a slightly different set of variants. So the moment you bring in more genomes, the dataset size expands, and with about 100 individuals, you can capture variations at around 0.6% of the genome, and this can give you quite reliable information on many aspects of evolutionary history. So as a comparison, while a single or a couple of genome gives you a low resolution picture, with population scale data, let's say 20, 50 or 100, you start getting high resolution pictures, and the more genomes you sequence, higher is the resolution of evolutionary history that you get. As you can see that the number of variants detected also shows quite a wide range of variation. So for an individual it varies between 4 to 5 million, whereas at the population level it varies between 17 to 23 million. So what is the reason for this variation? So this variation is actually due to differences in ancestry of the particular individual or of the population. So as this plot from the 1000 Genomes Project study shows, here the number of SNPs in each individual is represented by a dot, and the same population is shown in dots of same color. African genomes on an average harbor almost 25% more variants compared to non-Africans. So why do Africans harbor so many more variants? So this figure from Karina Slebush's 2017 paper demonstrates why this happens. So as you will note here, all the initial divisions, that's the split of ancestors of Khoisan hunter-gatherers, followed by the divergence of ancestors of rainforest hunter-gatherers. And here you can see the y-axis actually is time, so there you can see that some of the splits are really ancient. So finally the split of the East and West Africans, which is also probably around 100,000 years old, all happened within the continent. And it is estimated that all the current non-Africans are related to the East African branch, as the out of Africa migrations originated from this. Therefore, barring this, all branches and all other ancient lineages remained and flourished only in specific regions of Africa, which enabled them to be so diverse. And despite this immense diversity, African genomes are so far the least studied among all populations. It's very difficult to estimate these numbers, but a reasonable estimate suggests that of the first 100,000 genomes that have been sequenced, less than 5% were Africans. And although projects such as StopMed are trying to bridge this gap, the genomes included in these studies are predominantly from African diaspora, and as we know from our knowledge of history, these diaspora populations represent only certain African geographies and ancestries, so they are not completely representatives. And even among the genomes that were sampled from the continent, as the slightly dated map of population scale WGS datasets depict, there is a clear bias towards certain geographic regions and huge gaps on the map. And only a few of the 2000 or so ethno-linguistic groups have any representative genomic data. So there is definitely a lot missing here. And the scenario is equally or even more bleak if we look into disease genetics data. Despite continental Africans comprising about 15% of the world population, less than 1% of participants in disease genetic studies are continental Africans. So the genesis of the studies that I'm going to talk about today lies in the Human Heredity and Health in Africa, or H3Africa Consortium's decision to do something about this gap. So for those who are not familiar, H3Africa is a National Institute of Health, Wellcome Trust, and African Academy of Science funded project to establish genomic tech and expertise across Africa. This is a $180 million initiative involving 50 projects, 500 researchers, and spread across 34 African countries. So today I would start with how our WGS study informed our understanding of diversity, selection, and migration. And as many of you know, this was published in Nature last October. And given the current standards of whole genome sequencing projects, a study based on 426 genomes is indeed quite modest. However, what makes this study special and perhaps enabled us to garner so much attention was the fact that the reported whole genomes were from Africa. So the geographic spread of the consortium that we saw a couple of slides ago enabled us to strategically prioritize 323 samples from 8 countries, which could fill in some of the major gaps. And these samples were sequenced at 30x or higher depth. We also included 212 individuals from 4 other countries that were sequenced at medium coverage by another project within the consortium. And after quality control, we ended up with a data set that consisted of 40 million variants and 426 individuals. And this represents 50 different ethno-linguistic groups. So among the 40 million variants that we identified in our study, almost 8%, that's 3.4 million variants, were novel with respect to variant databases and previous sequencing efforts. So this was indeed quite high. And to unpack how ancestry and geography are reflected in the novel SNV content, we compared the novel SNV counts normalized to 24 individuals per population groups, as you can see in this plot. And although genomically speaking, Nigeria is the best studied African country, the Berome from this country, which was which is shown here, was found to have a large proportion of novel SNVs. And this kind of emphasizes that the current Nigerian groups are not only poor proxies for the whole of Africa, but maybe poor proxies for Nigeria as a country as well. However, the novel SNV represented here was pale in comparison to what we saw in Botswana, and admixtures plots for Southern Africa show that the Botswana have significantly high Khoisan ancestry, here shown in green, and we also found a strong correlation between the proportion of the Khoisan ancestry and the number of novel variants in an individual. So similar correlations were also observed for other ancestries such as North Africans, rainforest hunter-gatherers, and so on. And this kind of shows that there is a tremendous potential for novel variant discovery in African populations if the populations are chosen so as to bridge geographic or ancestral representation gaps. So we then surveyed the frequencies of select well-described disease-associated variants, and some of these are depicted in this picture. So RS334, which is the common sickle cell disease mutation, locus that protects against malaria mortality, was found at typically high allele frequencies in malaria endemic West and East African populations, and this was expected. However, what was interesting was that the HBS showed widely divergent allele frequencies in two populations from Uganda, with the Bantu-speaking UBS population having one of the highest frequencies, that's around 20%, whereas this allele was not observed at all in the Ugandan Nilo-Saharan population. And while this was due to major ancestral divergences, as these PC and admixture plots show, these extreme differences underline a critical point that for Africa, estimates of disease susceptibility or genetic risk needs to be much more fine-grained, and a single estimate for a country might often be misleading. So next, we tried to assess the contribution of natural selection to the genomic landscape. And for this, we employed a very widely used estimate known as composite likelihood ratio, or CLR, in each of the population. And this method is actually, using frequency of mutations in a genomic region and using a quite complex computational methodology. It assesses whether a region has undergone selection or not. And this circular Manhattan plot shows the selection signals, or CLR scores, across 10 kb windows, and this is like the whole genome, and the concentric circles are for Botswana, Zambia, Cameroon, Nigeria, Benin, and Ghana and Burkina Faso, moving from outside to inside. As this is quite difficult to see, you have to take my word for it that we identified 107 signals in these 6 populations, and 62 of these were in novel genes. And as expected, a large proportion of the novel genes were involved in immunity, DNA repair, and metabolism. So immune genes are somewhat common in any selection scan, and several signals for immunity to protozoan diseases such as APOL1 and HBS are very well known in African populations. However, what was surprising was that a substantial proportion of immune genes detected in our scan were related to viral immunity. And this suggests that viruses might have also played a role in shaping genomes, at least in some parts of Africa. And although almost half of these novel signals were seen in 2 or more populations, several were seen, as you can see in this rectangle, that they were either population or geographic region specific. Moreover, for some of the well-known signals such as FOXP2 and APOL1, they showed very high variability in selection strength across the groups studied. So, given a recent shared history and geographic proximity of some of these populations, these differences were intriguing, and it would be a great topic for in-depth investigation as larger WGS datasets become available. So, moving to migrations, so long distance migrations have played a pivotal role in shaping the genomic landscape of the African continent, especially in the last few thousand years. And this is mainly by bringing ancestral populations inhabiting different regions of the continent in contact with each other and resulting in this characteristic distribution of linguistic groups. And there are several important migrations that shape African diversity. The most dramatic of this is perhaps the Bantu expansion, a series of migration that initiated around 4,000 years ago from somewhere in modern day Nigeria and Cameroon to vast areas across sub-Saharan Africa. And it is due to this migration that one of the Bantu family languages are the major languages of almost all sub-Saharan African countries. However, the exact sequence, timeline, and route of this migration is on one of the most debated topics in African archaeology, linguistics, and perhaps also genetics. So the PCA of our data, that when it was integrated with data from external sequencing studies, looks like this. And as has been observed for similar populations in previous studies, the first principal component, which is along the x-axis, separated the East Africans from other African populations. The second principal component placed the remaining populations along a line from west to south, and it's kind of like shows a very strong correlation with geography. So the individuals from Cameroon, DRC, and Zambia localized in proximity to each other and forms a somewhat independent Central African group. And here you can see the populations from Zambia kind of localizes in the crux of this line from west to central and then to south and east. And this unique localization of this group in the PC made us suspect that it might have interesting connections with other neighboring groups and perhaps maybe to Bantu migration as well. So the most current genetic database model is from a study by Patan and colleagues published in 2017, and it estimates a southerly movement from Cameroon to Angola before dispersal into south and east. And the inclusion of populations from DRC and Zambia provided us the opportunity to explore the potential role of these countries in the Bantu migration route. So when we looked at sharing of shared genomic segments, that's IBD sharing, we saw that the sharing with Southern African Botswana was greatest among the Zambians, and again all the East African Bantu speakers from Uganda were also found to show highest genetic affinity with Zambia among all the Central and South African populations. And we performed several additional analyses using admixture modeling, and all of them support the view that Bantu migration moved south of rainforest to present-day Angola and then perhaps moved eastward, and only after reaching Zambia the eastern and the southern streams separated out. So as you can see that this study has a lot of population genetic insights, and there was another utility which this genetic data kind of served. And as we all know that despite the plummeting cost of sequencing, the relatively cheaper price, ease in data handling and storage still makes genotyping array very widely used in disease genetic studies. And also much of what we know about population genetics comes from array-based studies. However, arrays designed on the basis of European datasets was long known to perform suboptimally in African populations. And as the major aim of the H3Africa was to conduct GWASs, the consortium decided to design its own Africa-centric array, and The novel genomes, along with public datasets, contributed to the design. So a key feature of this array was that in addition to 1.5 million SNPs from existing libraries, which are called beat pools, the array contained around 700,000 SNPs that were specifically inserted to better represent African LD, and there were a lot of functional variants which were also targeted to capture the SNPs that might be relevant for African GWASs and other studies. So this array has been used to genotype more than 50,000 Africans to date. And in the second part, we will visit a recent study that was based on data that was genotyped on this array, and we will see how it enabled us insights into the final stages of Bantu migration and the peopling of Southern Africa. So going back to Bantu migration, as the migration moved south of Zambia, something very interesting happened. So the migrants came in contact with Khoisan hunter-gatherers, which represents one of the most ancient and genetically diverse human lineages. And this was followed by a gene flow from Khoisan into Southern African Bantu speakers, which makes them very unique and perhaps uniquely diverse among all Bantu speaking populations. So the Bantu speakers just from South Africa speak several distinctive languages such as Zulu, Swana, Xhosa, as you can see in this tree. However, whether these linguistic differences are paralleled by genetic differences was not known. And although one of our earlier work hinted at this possibility, it was a very limited study based on a few samples and just two ethno-linguistic groups, so nothing was conclusive. So employing the Avicens dataset that included 5,000 participants from all major South African ethno-linguistic groups, we investigated whether there is a correspondence between language and genetics. So as you can see, the distribution of 9 major ethno-linguistic groups on the map as well as the language tree. Now the genetic data shows, as this you can see in this PC, a separation among the 3 major ethno-linguistic divisions, that Sotho, Swana, Nguni speakers and Songa. And not only that, if you look carefully, you would see there is a correlation between localization of these groups on the map and their localization on the PCA, showing that there is not only a population structure within the ACB, but the structure correlates both with language and geography. So key components of the history of a population are often reflected in how the size of the population has changed. For example, a population that's affected by adverse climate change or a disease would have undergone a reduction in population size. On the other hand, exposure to new resources or cultural changes such as adoption of a new farming technique could increase the population size dramatically. So we used a program IBDNE, which based on this premise, and it tries to investigate the changes in effective population sizes in last few hundred generations. So in this plot, we have summarized the population size changes in ACB groups in the last 100 generations. And as you can see in this figure, that all ACB groups are very similar for the initial 60 generations, and only then the individual groups began to diversify. And this matches with the generally accepted model of Bantu expansion, which suggests a sharing of a common ancestor by all present ACB groups until this time range. And any variation profile of the Nguni speaker, that's Zulu and Xhosa, was found to be similar but different from all other ACB groups. Similarly, the Sotho Swana also kind of shows a distinctive and characteristic profile, suggesting that the population structure that we observed has some level of correspondence to demographic history. So to delineate the timeline of the migration of Bantu speakers within Southern Africa, we dated the admixture between Bantu speakers and case source populations for each of the ACB groups using various techniques. So in this plot, dates based on fast globetrotter are shown in red, and those based on Mulder are shown in blue. And our dates show an overall correspondence with previous studies, but provides a more granular estimate of these admixtures. And the Songa shows the oldest admixture dates, which is around 45 generations ago, while admixture dates for other groups such as Swana and Khoza were around 25 generations ago. And these dates are supported by archaeological evidences which indicate that Songa might have indeed arrived before the Nguni speakers. So this kind of shows a model of Bantu speaker expansion within southern Africa to be a complex process which could have either involved multiple waves of migration or some form of punctuated expansion. So as we performed this analysis for South Africa, for this review, we tried to expand it, this kind of estimates for a wider geographic region. And for this, we just used available literature, and we faced a major challenge. As you can see here, each study out there has a different genotyping array that was employed. There were differences in dating methods and the Khoisan proxy data sets. So all these factors can affect the estimates of admixture dates. So we have a situation where we have substantial data to provide a decent timelines. However, as these studies were conducted in kind of methodological silos, unless we integrate them and homogenize them, we cannot do that reliably. So the collaboration within African researchers is required to use datasets in such a way so that we can homogenize them and generate both Pan-African and regional insights. So to summarize, the fruits of H3Africa Consortium can be broadly divided into three categories. Firstly, to bridge the gaps in publicly available whole genome data, we have been strategically sequencing hundreds of whole genomes at both high and medium coverage. So some of these are presented in this talk, and many of them are to be published soon. The consortium based on this data has generated an Africa-centric genotyping array, and we are busy integrating genomes into an imputation panel that would enable better imputation of continental African data sets. And the data generated using H3Africa genotyping array has not only enhanced our understanding of complex diseases by means of GWAS, but also leading to major leaps in our understanding of population structure and demographic history of specific African regions. And all these data sets are being made available via global repositories such as EGA, as well as local databases aimed at enabling African geneticists and bioinformaticists to be able to make use of these for their own research. So finally, I need to emphasize that I'm presenting all this work on behalf of three wonderful teams, and all the credit goes to these teams. And finally, sincere thanks to the funders who made all this possible, SBIMB, my research home, and the organizers for this opportunity, and all of you for your kind attention. Thank you.
Presentation. I think we'll take the questions at the end of the session because we're very short in time. So if I may present now Dr. Nadine Musambo. She specialized in ophthalmology. She did an International Council of Ophthalmology Fellowship in Pediatric Ophthalmology and Strabismus in Nigeria. Currently she participated in nanopore sequencing workshops of genetics unit at the University of Kinshasa, Democratic Republic of Congo. She was co-investigator in a population-based study on blindness and visual impairment in children in Kinshasa, and her field of research are uveitis and inherited retinal disorders. She's currently conducting a study on retinitis pigmentosa with the Center of Human Genetics at the University of Kinshasa. and she's Deputy Secretary General of the Congolese Society of Ophthalmology and member of the French Society of Ophthalmology. She's going to present us about phenotype-genotype of inherited retinal disorders in the Democratic Republic of Congo. Please go ahead, Dr. Nadine.
Hello everyone. I am Nesrine Mezzi. I'm PhD student at
Nesrine.
Pasteur Institute in Tunis. I'm supervised by Professor Sonia Abdelhak and Dr. Lilia Ramadan. Today I have the pleasure to present to you my work, which is entitled Spectrum of Genetic Diseases in Tunisia, Current Situation and Main Milestones Achieved. Diseases are defined differently from one organization to another. They imply great global burden and a significant challenge for health systems resulting from unmatched patient needs and absence of equal access to diagnosis and treatment. Nearly 6,000 rare diseases have been identified around the world. Only 5 of them have an FDA approved treatment. Approximately 72% of rare diseases
So it's Nadine who's going to present now. Dr. Nadine, would you go ahead, please?
Yes, it's Lusambo. Yes.
Dr. Lusambo, please. Dr. Lusambo?
Yes,
No, she's there. Just we need her camera on. Okay, great. Please go ahead.
presentation. The name of my presentation is Lusambo NN.
Vous êtes sur la deuxième dia. Entrez au début.
Au début,
Là,
hein?
elle est là où il y a la présentation. Revenez.
Yes.
Mettez le micro, ne parlez pas.
You're not ready? I go ahead? Okay, thank you. Sorry for this technical problem with Dr. Lasambo, so we're moving with Dr. Nassim Metzi. Dr. Metzi is a student of a close friend from North Africa, Professor Sonia Abdelhak and Dr. Lilia Ramadan. She specialized in the field of cellular molecular biology and biochemistry. She had her master's degree in updated databases of Tunisian genetic diseases. She's analyzed exome data aiming to identify deleterious variants in Tunisian populations, which I know the size of the genetic disorder studying. She acquired competences in bioinformatics, especially next generation sequencing data analysis. Now, this project, she's conducting a multidisciplinary investigation of ultra-rare genetic diseases in Tunisia. She has been implicated in facilitating the African genomic medicine training. She is currently involved in the African Precision Medicine Portal project. Dr. Metzi will present about the spectrum of genetic diseases in Tunisia, current situation, and main milestone achieved. Please go ahead, Dr. Nassim.
Hello everyone. I am Nisreen Mizzi. I'm PhD student at Pasteur Institute in Tunis. I'm supervised by Professor Sonia Abdelhak and Dr. Lilia Ramdan. Today I have the pleasure to present to you my work, which is entitled Spectrum of Genetic Diseases in Tunisia, Current Situation and Main Milestones Achieved. Diseases are defined differently from one organization to another. They imply great global burden and a significant challenge for health systems resulting from unmatched patient needs and absence of equal access to diagnosis and treatment. Nearly 6,000 rare diseases have been identified around the world. Only 5 of them have an FDA-approved treatment. Approximately 72% of rare diseases are attributed to genetic factors. Genetic diseases are early onset and life-threatening conditions. These diseases particularly concern the Arab world and several African countries. The spread of genetic diseases among these communities, especially rare conditions, were largely explained by socio-cultural factors. Indeed, highest rates of consanguinity with an extreme prevalence of first cousin unions were described across Africa and Arab world. Tunisia is a middle-income African country. It has been inhabited since the Paleolithic period. Berbers, also called Amazigh, are the indigenous people of Tunisia. Through its history, the Tunisian population has witnessed successive invasions and several migratory flows during prehistoric and historic periods, thus shaping its genetic landscape. Similarly to other Arab and African countries, familial and geographic endogamy is a deeply conserved rule in Tunisia with the rates ranging up to 38%. In this study, we reported the preliminary spectrum of genetic diseases in Tunisia. Of public health in Tunisia, as their spectrum encompasses 346 genetic disorders. In the present work, we performed systematic reviews of the literature relative to genetic diseases in Tunisia in order to provide a more recent spectrum of these disorders in our population, identify the type of potential health interventions that could be proposed for this condition in Tunisia, and to shed light on challenges that still exist. A comprehensive review of published data was conducted mainly using MESH and PubMed databases. The list of genetic diseases was obtained after querying the MESH database using the keywords genetic diseases. The query results were further combined to additional keywords Tunis, Tunisia, and Tunisian using the PubMed search tool. We focused only on papers published from 2010 to August 2021. Retained papers were included in the meta-analysis and were curated manually. They were completely screened for disease name, OMIM ID, inheritance mode, associated gene, germinal mutations, and epidemiological parameters. We also searched the same information from grey literature. From the selected documents reporting on genetic diseases in Tunisia, a recursive search for additional documents were performed using author names of referring physicians and researchers in Tunisia. Then we used the 10th version of the World Health Organization International Classification of Disease in order to provide a clustering of genetic diseases in different pathological groups. Data have been stored in a local database for subsequent analysis. Let's move to the main results of our study. In this study, we present the actual situation of genetic diseases in Tunisia. Compared to what has been published, 243 additional genetic conditions have been identified. Thus, the total number of genetic diseases in our country amounts to 589 genetic diseases, of which 41.3% come from the present data mining. We opted the International Medical Classification of genetic diseases described in the Tunisian population. Therefore, such classification revealed that congenital malformation, deformations, and chromosomal abnormalities are the most commonly reported. Endocrine, nutritional, and metabolic diseases are the next most frequent group of diseases, followed by diseases of nervous system. Genetic diseases derived from this analysis, autosomal recessive diseases are the most frequent, followed by autosomal dominant disorders. X-linked conditions were encountered at 5%. 61% of the 589 genetic diseases have a known molecular etiology. Molecular investigation of genetic diseases is mainly conducted using Sanger DNA sequencing in Tunisia. However, since 2013, next-generation sequencing has become more accessible to the scientific Tunisian community, leading to the identification of the molecular basis of at least 45 Mendelian diseases described among Tunisian patients. Target gene sequencing is the main genomic tool used to unravel the genetics underlying rare Mendelian disorders in Tunisia. Heredity is defined as the expression of another disease in addition to the primary disease. This phenomenon has been reported among Tunisian families. In our updated data, we have identified 14 co-occurrences of multiple genetic diseases with a consanguinity history among 6 of them. Both familial and individual comorbidity have been identified. They are either with the same mode of transmission or not. Local phenotypes highlight the genetic disease spectrum among Tunisians. Shannon-Dorfman syndrome, palmoplantar keratoderma, and Parkinson disease were described in Tunisian patients with unusual presentation. Then, few patients present undiagnosed phenotypes suggesting either new clinical entities or atypical expression of rare or ultra-rare disease. For epidemiological parameters, we focused on the frequency of the whole reported disease spectrum by targeting either the prevalence or the incidence. We noticed the paucity of such epidemiological data, as only 27 genetic diseases described among Tunisians have defined prevalence and/or incidence. For the remaining diseases, only the number of studied patients and/or families is reported. 223 genetic diseases were described to be screened among newborns and/or at-risk individuals in the world, of which 31% belong to the recommended uniform screening panel. Others are diseases that meet the first 6 criteria proposed by AfSHG for expanded carrier screening panel. Of this disease list, 49% are described among Tunisian patients, representing 19% of genetic disease spectrum in Tunisia, which are therefore potentially target for intervention. Collected data are being integrated into a database under construction that will be a valuable decision-making tool. In this study, we attempted to provide a comprehensive and up-to-date overview of the genetic disease spectrum described among Tunisians. A significant rise in the number of studied disorders were observed in this population. In the period of 11 years, the number of reported genetic diseases increased from 346 to 589. This highlights the availability of better opportunities to perform molecular diagnosis. This suggests also a remarkable progress in Tunisia, particularly through the development of biomedical health research activities, building capacities in this field, and the possibility of international collaborations enabling the use of high-throughput sequencing to overcome the scarcity of NGS platforms in our country. Here in the recent spectrum of genetic diseases. Most of these diseases identified among Tunisians are autosomal recessive. The frequency of autosomal recessive disorders is still high and could be explained by the elevated prevalence of endogamous mating. The clinical heterogeneity could be challenging in molecular diagnosis. Such characteristics render the diagnosis difficult and could lead to misdiagnosis in developing countries such as Tunisia. Screening for founder mutations, especially among endogamous populations, is a cost-effective molecular diagnosis strategy, but it may be inefficient when applied to atypical rare phenotypes and comorbidities. Another impact of elevated rate of consanguinity and inbreeding is the co-occurrence of two or more phenotypes within the same family. Comorbidity, described particularly in inbred populations, makes genetic counseling and prenatal diagnosis challenging. Taking into account the challenge in diagnosis heterogeneous genetic conditions and phenotypes, there are more complex Another impact of the elevated rate of consanguinity and inbreeding is the co-occurrence of two or more phenotypes within the same family. Comorbidity, described particularly in inbred populations, makes genetic counseling and prenatal diagnosis challenging. Taking into account the challenge in diagnosing genetic conditions and phenotypes that are more complexified by comorbidity in highly inbred populations, NGS seems to be an efficient and rapid genetic diagnostic tool that has marked a shift in genomic medicine in Tunisia. The dearth of data concerning genetic conditions epidemiology in Tunisia suggests that the health burden of such diseases is significantly underestimated in our country. This indicates the necessity of comprehensive epidemiology and patient registry implementation, which could highlight the genetic diseases burden in a population and serve as a worthwhile resource for improving the quality of health care received by patients. From public health perspective, experts and policymakers have opted that newborn and career screening should be conducted as a state-run program. This will allow to an early detection of genetic conditions and congenital disorders, leading to reduced health disparities and less death at a young age. Infant mortality is an indicator of states' health, and reducing this rate is an explicit goal of sustainable development. In addition, reducing genetic conditions expression, particularly those recessively inherited, is a salient issue in several African countries. Targeted screening of prevalent genetic conditions in Tunisia could be a cost-effective alternative in our country, since they have been associated with founder mutations. This could allow to early detect newborns with these conditions and also to identify at-risk individuals. Molecular data availability on relatively frequent genetic rare diseases in inbred populations could be an ideal way that boosts current research focusing on targeted therapies, including gene and mutation-specific therapies, hence leading to the implementation of precision medicine. Several diseases identified in Arab and African communities are subject to targeted therapy based on their genetic information like Bloom syndrome and primary hyperoxaluria type 1. There are several strengths to our study. One of them is that a better knowledge of the genetic disease spectrum in Tunisia is useful for this country as well as for other countries in order to improve the diagnosis, to reduce misdiagnosis, and therefore will have earlier management. There are several strengths to our study. One of them is that a better knowledge of the genetic diseases spectrum in Tunisia is useful for this country as well as for other countries in order to improve the diagnosis, to reduce misdiagnosis, and therefore patients will have earlier management. Molecular and genetic data for these diseases are becoming more widely available through the development of biomedical health research activities and the access to high-throughput next-generation sequencing. Although the Tunisian population still faces challenges, especially in the case of unidentified phenotypes, which delays the appropriate patient management. These challenges include a paucity of diagnostic facilities, limited access to technological advances. Such issues may be overcome by creating diagnostic referral centers that leverage some technology like high-throughput sequencing for genetic diseases elucidation in the country. The specialized structures could potentially offer accurate molecular diagnostic services at a lower cost and allow faster, efficient patient management. Also, patient management should include multidisciplinary team investigators working in a well-defined care pathway. Researchers are needed to accurately set the epidemiological data of genetic diseases, aiming to implement better and appropriate healthcare measures. Patient support groups have a significant role in promoting patient voice and influencing the decision makers. Finally, I would like to express my sincere gratitude to my supervisors, to also to all the members of the project H3 Africa and H3A Bionet, to the African Society of Human Genetics, and to Tanzanian Society of Human Genetics. Thank you for your attention.
And because the time is so tight, we're moving ahead. Please, Dr. Maso, are you ready? Lusambo, sorry, Dr. Lusambo, are you ready? Yes, please, go ahead.
I would like to thank the organizer of the meeting to give me the opportunity to present. I am Dr. Nadine Tiangani Lusambo. I am a consultant ophthalmologist in the teaching hospital of Kinshasa and a senior lecturer in the medical school of Kinshasa. I'm presenting you our work on phenotype and genotype of inherited retinal diseases in the Democratic Republic of Congo. This is the plan of our presentation. Inherited retinal diseases are a heterogeneous group of disorders that cause incurable visual loss, mainly due to the dysfunctions or degenerations of rods and cone photoreceptor cells in the retina. Dozens of different diseases are included, with wide and sometimes overlapping spectrum of phenotypes. The age of onset of RAD is highly variable, ranging from childhood to mid-adulthood. An early clinical feature of RAD is night blindness, often starting in adolescence, followed by progressive loss of peripheral visions, and in many cases culminating in the loss of central visions and complete blindness in midlife. Ophthalmologic hallmarks of the disease include an abnormal fundus with bound spicule pigment deposits, attenuated retinal vessels, chorioretinal atrophy, and a waxy optic disc pallor. Electroretinography often reveals severely reduced or non-detectable responses. Patients can also present reduced visual fields. RAD can be subdivided in two types: non-syndromic RAD without systemic abnormality, syndromic RAD, where retinal degeneration is associated with other symptoms due to systemic diseases. There are two most frequent pathologies: The Usher syndrome, characterized by early onset of congenital neurosensory impairments, followed by the development of RAD, and the Barden-Biedl syndrome, characterized by RAD, polydactyly, obesity, renal abnormalities, hypogenitalis, and mental retardation. Many other complexes and pleiotropic conditions include RAD as a component. The inheritance patterns of RAD can be classified as autosomal dominant, autosomal recessive, X-linked, diagenic, and mitochondrial. The autosomal dominant form concerns 30 to 40% of cases. Autosomal recessive is about 50 to 60% of cases, and the X-linked form concerns 5 to 15% of patients, and is the most severe subtype of the disease. Rare cases of RAD presenting mitochondrial or diagenic inheritance or uniparental disomy have also been described. A number of cases cannot be classified due to the lack of family information and are simply referred to as simplex. Overall, more than 200 genes are associated with RAD. The non-syndromic form, mutation in more than 15 genes, are known to cause the disease, and nearly 3,100 mutations have been reported in these genes. Mutation in 12 genes cause Usher syndrome, and in 16 genes are associated with Bardet-Biedl syndrome. Together, these two diseases account for 1,200 pathogenic mutations. The relative frequencies of the different genetic forms of RAD vary in different populations throughout the world. In Africa, there were some studies carried out in Nigeria, South Africa, Egypt, Algeria, and Tunisia. No study is available in Central Africa, and very little information exists on the phenotype and genotype of RAD in Central Africa. The purpose of our study was to identify the genes and mutations responsible for the RAD in the Congolese population. The study was carried out in Kinshasa, the capital of DRC, in two hospitals, in the teaching hospital of Kinshasa and the Saint Joseph Hospital. Inclusion criteria of our patients were a clinical diagnosis of RAD and to agree to participate in the study. We included 7 indexes from 7 families. Each patient underwent a standard clinical examination with a dysmorphism evaluation and a complete ophthalmologic assessment. A venous blood samples was taken for all the patients. The DNA samples were extracted in the Center for Human Genetics of the University of Kinshasa. Then the samples were sent in USA, where the clinical whole genome sequencing were performed in the clinical services laboratory of the Illumina through the Illumina High Hope program. Our series included 4 men and 3 women. The age of our patients ranged between 27 to 77 years old. We have 2 familial cases and no dysmorphic cases. About ophthalmic finding, 5 patients complained of decrease of visual acuity, and 3 of them had hemerolopia. In fundus examination, bone speckles were found in all the patients. 3 patients had a tightening of retinal vessels, and 2 of them had a retinal pigmented epithelium atrophy. In the first family, we have a sporadic cases, and the clinical whole genome sequencing did not allow us to identify a causal variant for the RAD phenotype. Genomic data will be reanalyzed in 6 months' time hoping to solve this case. In the second family, the mother and 3 daughters were affected. The clinical whole genome sequencing identified 2 novel mutations in the ADAM9 genes for the 3 girls, including a stop mutation marked with a red asterisk, classified as pathogenic, and a small intragenic deletions marked with a green asterisk, which is classified as likely pathogenic. Interestingly, the affected mother is heterozygote for the deletion only. In the absence of the father, we assumed compound heterozygotes for these 2 women and suspect that the affected mother could reflect a novel inheritance form of ADAM9 related corneal dystrophy, similar to mixed inheritance observed in RP1 associated IRD. In family 3, in this woman, the clinical whole genome sequencing identified a novel stop mutation in the RP1 gene in heterozygous state. This mutation is classified as likely pathogenic. The other family members are reportedly unaffected and were not available for testing. We assumed causality of this variant and returned the diagnosis of retinitis pigmentosa type 1. In this man, the clinical whole genome sequencing identified 2 novel heterozygous variants in the MERTK genes and a missense variant. These variants are classified as a variant of uncertain significance and likely pathogenic respectively. The other family members are reportedly unaffected and were not available for testing. We assumed causality of this variant and returned the diagnosis of retinitis pigmentosa. In family 5, the clinical whole genome sequencing of this man identified 2 heterozygous variants in the CYP4V2 genes. Both are novel missense variants. These variants are both classified as variants of uncertain significance. The other family members are reportedly unaffected and were not available for testing. We are performing now the phenotype reserve to validate this diagnostic. In family 6, the mother and her first son are affected. Clinical whole genome sequencing identified a pair of variants involving the USH2A gene and a 8 kilobit deletion of chromosome 1, which is not found in the proband's mother, and a stop-gained variant that is present in the mother. Both are de novo and are classified as pathogenic. Variants in USH2A are associated with Usher syndrome type 2, which is consistent with the reported clinical phenotype. We assumed compound heterozygotes for this man, which is in consistence of a novel inheritance form of Usher syndrome. So conversely, for the mother, we carrier for the heterozygote SHA2, similar to mixed inheritance observed in RP1 associated IRD. In family 7, the clinical whole genome sequencing of this woman identified an homozygous missense variant in the HFT140 gene. This is classified as a variant of uncertain significance. We are performing now the phenotype reserve to validate the diagnostic. In conclusion, we report on the first cohort of African IRD patients investigated by clinical whole genome sequencing. Our results indicate that the spectrum of IRD causal genes is broad in Congolese patients. We have expanded the spectrum of causal variants for IRD These findings will be included in publications and deposited in ClinVar. We also raise questions on possible mixed inheritance for two hereditary genes previously constrained to autosomal recessive inheritance. This report demonstrates the power of clinical whole genome sequencing as a first-year test in individuals with a suspected genetic diseases in resource-limited African population, especially for genetically heterogeneous diseases where multiple cases of missing heredity have been reported. We are thankful to patients and their family for consenting for the clinical whole genome sequencing and giving the authorization to use the image. We thank the Illumina IOP program for donating the clinical whole genome sequencing and to the Illumina Clinical Lab for the analysis. Thank you to my supervisor, Professor Emelu Maka and Jehimu Kungu, and to my colleagues. Thank you for your attention.
Very interesting and informative presentation. It's interesting to see this data coming from Central Africa. If we can move quickly to the questions and answers, because I have an ultimatum. So I will start with the very few questions coming to Nasreen. Two anonymous questions that can be compiled together. They're asking about, do you think is affecting the study of genetic disorders? And this is one, and the second is, what is the size, or do you think the size of the sample that you need to study genetic disorders? Do you think a small sample in this consanguineous background is better, or do we need a large sample to analyze? Nasreen, please.
Yes, I can't hear you well. Could you please repeat the question?
Yes. The first question, the second question, let's start with the second one. How big the sample you need? Do you think a small sample size is better to study in this consanguineous background in Tunisia? Can you hear me well? Nasreen?
Yeah, Prof, your voice is fine. I think it's some right.
Thank you. I thought I have a problem. Okay.
No.
So, okay. So we move to. Okay. Let's
okay,
yes,
I'm
there she is.
with you.
Yes.
Sorry.
Okay, Nasreen. Please go ahead. Did you hear the question?
Okay. I hear you.
Heard it?
No. Sorry.
Okay. So interesting that family 2 had all know this is for, this is not for you. Just a second.
Okay.
Do you think consanguinity requires smaller sample size for your methods? Nasreen?
No, consanguinity is described among our population with high rates. So generally, genetic diseases are described among families with consanguinity or endogamy. Is it?
Okay, and Professor Raj is asking you, hi Raj, is it, he's keen on knowing what are the number of medical geneticists and genetic counselors in Tunisia? The number of genetic counselors in Tunisia.
Okay. In Tunisia, we do not have this field. We do not have genetic counselor as a specific field as a defined field in our country, because we do not have the well-defined path of genetic diseases. Yes.
We do not have what the number of medical geneticists? Do you have a definite number of medical geneticists in Tunisia? Difficult to
Right.
answer, huh?
Yes, it is difficult to answer, but we have yes, but we have four referral centers in our country, which is insufficient to well diagnose, elucidate the molecular etiology or clinical manifestation of these diseases.
Okay, thank you very much.
You are
Dr. Losambo, interesting that family 2 had all the females with the disorder and no males had it. The mother and all the daughters. The D is the ADA M9 deletion, highly penetrant. Could there be mitochondrial DNA or other chromosomes involved? Dr. Losambo?
We think that this a novel, a new mode of we think perhaps that's a new mode of inheritance about the
New mode of inheritance? Or new mutation?
a new mutation,
New variant, yeah.
about the IRD, and we must do more explore.
Okay, okay, thank you very much. Now coming to Dr. Chaudhry with all the questions.
It can be a coincidence, but it could be also a.
Excuse me. It could be yes. It could be mitochondria. So, Dr. Chaudhry, it's anonymous speaker or someone who's asking the question. Great talk. The diversity of UNS and UBS is in Uganda is interesting. Number one, what do you think are the impacts on common complex disease genetics in that context? Let's take it one. Okay.
That's a great, yeah. So this particular population, we know what the story is, so we could go deep and we could mine that actually one of these populations probably migrated to Uganda. So these populations live in the same country right now. They have been separated by a bit of distance. I mean, like maybe a couple of 100 years ago. So that could explain why we are seeing this kind of in disease in spite of geographic region. So that's kind of answers. I mean, that's an easy answer, yeah.
And do you have other insights on Nilo-Saharan studies that have been explored today?
Yeah, so I think we haven't had a single Nilo-Saharan population. So for this particular study, we didn't have a lot of options, but we actually looked into Nilo-Saharan, how our Nilo-Saharan population clusters with other Nilo-Saharans, and we indeed found that there are very clear clusters with Nilo-Saharan. In some places, they have more Afro-Asiatic admixture. In some cases, they have slightly, in the rainforest, they have, some of these populations have rainforest hunter-gatherer. So, the Nilo populations are also incredibly complex, and that have exploded have all shown it, and I think we need bigger studies to really look at it.
Another question. Thanks for your comprehensive presentation. It's so exciting to see the ways H3Africa is expanding. This is a question we said to each other. The representation of all of humanity in genomics research. In what ways have the groups you work with collaborated with community engagement experts? This is a very interesting question, yes, and ethical experts.
Exactly. So I think for each, the whole genome study, what we did was, because all the H3Africa sites have their own community engagement and ethics engagement protocol. So in many cases, we kind of piggyback the existing samples. So we didn't really had a chance to go to the field or collect samples or be involved in that. So these were the samples that already collected. H3Africa, it's using H3Africa ethics and everything. For the Southern African study, we were actually a part of the collection at Abidjan. So there, yeah, we had like a very serious, and even after the study, I think Michelle Ramsey, she went to the particular sites, and we participants that what we found, and we're also kind of happy. So it's kind of the complete loop.
Yeah, you answered the second part of the questions about South Africa and how the political social landscape is different.
No, so I think exactly it's very different. So I mean, it's so different. Yeah, I mean, it's beyond. We can't completely admit that this is very complex.
It's Sigan Fatumo. She's asking about. Would you mind throw more light on the imputation panel you mentioned? Had this been assessed against other panels in the region? And top map?
Yeah. And so this is still kind of work in progress. Yeah. So we are, so this panel is kind of under development, and we have already got 6 versions of the H3A panel, and we are evaluating each of them. So I think I shouldn't be doing this right here, but I'm sure someone from this group or maybe us, we will answer this in a few months. But yeah, I get to that area right at this moment.
Okay, that is for the questions. Maybe I can ask my question now. Why is Northern Africa was not represented in your data? Although I know that Tunisia and Egypt are participants in the H3Africa Consortium.
Thanks. That's a great question. And I mean, I was expecting that someone asked the question, and I would break that we are actually planning the next phase of this study, which is going to be much bigger. And this time, one of our focus areas is North Africa. So we are really asking everyone in North Africa, whoever has access to samples. So you might even get an email from us saying that samples with consent so that we can partner together. But I think we completely understand that, and we understand North Africa, we are not going to complete the genetic map, and I think it's one of our focus areas for the next study. And hopefully that will be sometimes next year. So keeping fingers crossed.
Hopefully. Thank you very much, all the speakers, for very interesting presentations, and we're very sorry for the technical mishaps. Thank you very much. Okay, bye.
Thanks. Bye-bye.
Bye.